speak in this presentation about this segment of the arm of the activity that we have, which is to pure industrial pharmaceutics related work, which is categorized under the development of controlled or sustained release dosage forms for oral administration. Why metformin? Why diabetes? All of us knows that this is a hot topic. It's very important. Why? If you look at the numbers here, simply, which is these are numbers which are announced by the WHO regarding the 2,000 facts related to diabetes, as well as the projection for what's going to be the statistics in 2030. So you can see that, for example, the three leading countries, which are India, China, and the United States, if you look at the numbers, in 2030, they are predicting that the diabetes will be, there are going to be 79.4 million persons suffering from diabetes. And it's actually a progressing problem. This is as well, again, from WHO, which is comparing the incidence of diabetes in adults, comparing between developing countries and developed countries. And as we know, the incidence in the developing countries would be really much higher. And at the same time, you can see that this is also comparing the projection between 2000 and what's going to happen in 2030. Huge. We're talking about a serious problem related to diabetes. So, to sum up those, more than 220 million people worldwide will have diabetes. And in 2004, that's based on the WHO facts that was announced in January 2011, more than 80% of diabetes deaths occur in low and middle income countries, as I just showed, and the WHO projects that diabetes deaths will also double between 2005 and 2030. So we're talking about really serious health issue, and that's why I'm trying here to present why we are really going back to looking at diabetes, or keep looking at diabetes as really as an important target. Now, for me, of course, to get funded for the project, I have to fulfill the criteria for Qatar National Priority Program, but at the same time, the statistics that I'm showing here is actually applicable to all the GCC countries. So the figures here with regard to diabetes in Qatar, same thing or approximately the same in Saudi, in UAE, in the Middle East in general. So the latest WHO estimate, the prevalence of diabetes in Qatar in 2000 is around 38,000. That would really be at least to 88,000 by 2030. And of course, we'll say, well, what is 80,000? It's nothing. But this is for a 500,000 population. It's a huge. So that's the thing. We have to put it into perspective. And maybe this figure would really explain it more, because here we're talking about and I couldn't really get the statistics, but this is, as I said, in general, the newspaper lately announced that through the Diabetes Association, that the prevalence of diabetes in Qatar, which is, as I said, similar to the other Middle Eastern as well as mainly the Gulf countries, we're talking about 28%. That's huge. And part of it is due, of course, to the lifestyle, to the many other factors. Now, for this reason, when you look at it from industrial point of view, it's always better to look at the existing drugs rather than investing millions. And at the end, you don't know what's going to really be the outcome. So to maximize the therapeutic effect of the existing drugs would be really the main targets. And one of the best drugs used is the metformin. Why? This is a biguanide, which is one of the best highly prescribed antihyperglycemic used. This is one of the oldest and fastest or safest medications, and many, if not all, the diabetic type 2 people, patients would really use metformin as their first choice if they can. And one of the advantages is that it does not really induce hypoglycemia as other anti-diabetic agent, so there is no possibility of having a hypoglycemic shock with metformin. And it's also, which is good, the diabetic persons or diabetic patients will not really suffer from the weight gain. On the contrary, some people right now or nowadays, they take metformin low dose in order to have weight loss. And as well, it has no problems, or it's actually positive in case of the cardiovascular risk, especially when those who have dyslipidemia as well as insulin resistance. And this is why we picked metformin as our drug of choice, because it has some problems. It's well prescribed for diabetic patients. So what do we know from physicochemical point of view? Now to shift it to the pharmaceutic side. It's a weak base with a pKa of 12.4. It's highly water soluble. A drug which is crystalline in nature. It has a short and variable half-life. And I'm highlighting this in red because it says absorbed in the upper parts of the small intestine, which is the duodenum as well as the jejunum. And as a result of that, you would expect that a highly water-soluble drug and with good permeability, it should really have not really any problem with regard to the bioavailability. However, because of the site-specific absorption, which is in the upper part, it means that we have to retain it for longer time in the stomach in order to get absorbed well. So this would result in the fact that metformin bioavailability is around 40 to 50%. And this explains why those who are aware or familiar with metformin, we're talking about high doses. So the starting dose is 500 and gets up to 1,500 milligram. So this is all given in order to compensate for the low bioavailability. And again, as I said, it's because of the specificity of the absorption site as well as the high water solubility, which render it a little bit less bioavailable. And even in the industry, the powder of metformin itself has a problem of that it has poor compaction properties, which makes it a little bit difficult to formulate it, and that's why you'll find that the high doses of those are really bulky, because you have to put lots of the inactive ingredients in order to enable it compatible with the tablet press. So metformin is just like any other drug. When it's oral drug. So the drug has to go through the phases of, that we all know. So the tablet has to go through the disintegration first. It has to be in solution form with good biopermeability in order to be able to penetrate through the GIT to the bloodstream. If this process was not really completed well in the site of the best absorption site for the drug, you will end up with less optimal dose, as well as less clinical activity. So, many attempts, remember I said it's in the upper part of the small intestine, we have to keep it in the stomach for longer time. So now the research that is ongoing on having the metformin kept in the stomach so that it will be adjacent to the upper part of the GIT in order to get absorbed well in that specific site. One of the technologies is called the gastro-retentive swelling tablets, and I'll explain the concept in a minute, which is aiming to improve the bioavailability much, but the existing as well as the research have not really succeeded in exceeding the bioavailability more than 15%. So we're talking about still very low margin of success. The other strategy would be to use the hydrophobic or reported the hydrophobic triacetyl beta-cyclodextrins, which is used to develop sustained release carriers for highly soluble drugs. Again, still have short biological half-life, and the bioavailability was not really greatly improved. The floating microcapsules, pressed tablets, which is similar in concept to the gastro-retentive swelling tablets, which is to try to have the tablet floating. And as I said, I'll share with you the slide which describe the different or the two most successful mechanism, as well as the use of or design of the regular sustained release tablets in order to sustain the release of metformin. However, this was not really successful in retaining metformin in its sustained form in the place where it should be really absorbed. So Let's look at as well, there is the enteric coated tablets, but again, delayed the release, but did not really solve the problem with bioavailability and the site specificity of the absorption. So the floating tablets, or what we call the gastro-retentive, it's just depends, the principle of it is very simple. So you have to use what we call low density material in order for the tablet to float in the fluids in your stomach. Now, immediately, you will think of, okay, that's assuming that there are enough fluids for it to really float there and, to retain or to elongate the time for it to stay in the stomach in order to give the action. So you all and me knows that this will really depend on the gastro-retentive time and the transit time of the GIT, when is there food, is there not food, and what's going to be really the emptying time of the stomach. So this is going to really result in a huge variation. And this is the concept. So the floating tablet will stay there, it will swell, staying longer, and then it will sediment in the form of pellets, and then it will stick to the wall of the stomach. But again, there could be that the emptying could really happen during the first half an hour due to the fact that the gastric emptying was more active because of the presence of food and less fluids. So again, lots of variation in this. There's another technique or technology which really as simple as making a tablet, and this tablet will really swell to a huge size inside the stomach to the point that it will not really leave the stomach. And so it is really that you get this tablet. This tablet is going to really swell significantly, and when it tries to leave the stomach, it will stay there for a longer time till full degradation for the polymer takes place. And again, it will depend on the amount of fluids as well as the size of the tablet. Is it going to really, after swelling, will it be really enough to not to leave the pylori, or it's going to really stay in the stomach? Lots of variation. So, based on what we have there, we came with the idea of using simple, and usually in pharmaceutical industry, the simpler, the cheaper, the more successful. And I learned this from my presence with the industrial sector rather than in the research. In the research, we can imagine lots of things, and we have a huge imagination, but we publish 1 or 2 papers, but it does not really materialize into being marketed or being beneficial to the patient at the end. So based on this, we thought of the fact that we wanted to have a formulation that will be really enabling the metformin to stay in the stomach, increase the bioavailability, and at the same time having really lower dosing regimen and dose to the patient. So the simple idea is to use actually a hard gelatin capsule. And in that hard gelatin capsule, you would really use a hydrophilic polymer. You see, I have a special collection with PEG 35,000. I don't know why. But anyways, I'm giving here a range. And this represents a hydrophilic polymer. And what's going to do is that those type of the high molecular weight hydrophilic polymers will really, upon contact with gastric fluids, they will swell and form really hydrogel. And this hydrogel, the drug can really penetrate through the pores of that gel. So it will really act as the first layer of sustained effect for the drug. The second components or excipients would be the glycers, which are well known as hydrophobic polymers. These are triglycerides, and those will actually close, this is the hypothesis that we have, will close those pores and will act actually as a second layer for retaining or sustaining the release of the drug. At the same time, we wanted something to not only contribute in the sustaining the release, but also to be able to stick to the stomach wall. And this is what we use the mucoadhesive, usually. It's used in the mucoadhesive tablets. We've used the gastroadhesive polymer, which is the carpomer. And we are actually, by this, we are hitting two birds in one stone. First, carbomers are well known that they are really components which can really control as well as sustain the release of the drug. And at the same time, they will stick to the wall of the stomach, hopefully. This is all hypothesis, but we have to prove it, and inshallah I'll show it to you. So with the experimental design that we had, and I don't want to go to the statistical point, we've prepared a series of formulations. And what you see here is the code for each formulation from F1 to F14. And I hope I'm going to simplify it as much as I can. And these are actually the main ingredients. So, and we chose actually the dose of 500 milligrams as the starting dose for the metformin, because the target at the end is that we don't want people to take lots of, our high dose. Half of it is going because there is no bioavailability. So that's the main target. So that's the base dose of the metformin. So MH is metformin. Juliucer 5013 is, and Juliucer 4414 are the hydrophobic excipients. The PEG 35K and PEG 400 are the hydrophilic ones, and the carbomer is the gastroretentive or gastroadhesive polymer. And what we've done is that we've fixed the fact that metformin has to be in 500 milligrams. So this is the dose that we are intending to. And then we've also fixed in the first formulation Juliucer 5013, only the hydrophobic one, as the component. And then everything else that we are adding from the other excipients would really be on the expense of the Juliucer. So for example, if we are adding here carbomer is 0.05, you'll find that Juliucer ratio is 0.95. In this case, where we have lots of other ingredients, we have 500 milligram of the metformin with 250 of Juliucer, 100 of Juliucer 4414, 100 for PEG 35,000, and so on. So we started to look at what are really the effect of this is going to be on the release of the drug, and eventually to pick the right formulations to advance to the in vivo study. So before we go there and we go far, let's investigate what's going on with the existing products in the market, which already we are claiming or we are accusing them of having low bioavailability. So when you do the in vitro dissolution testing, so we took the glucophage, which is the brand name of metformin in the market, and it's available in the instantaneous release as well as the extended release. Of course, for us, the XR is more of interest to us because we are designing a controlled release capsule, which should compete with this one here. And as you can see, as expected, the IR, and this is, as I said, the commercial one, the IR within half an hour, it released all its content as expected, as an instantaneous release tablet. While for the XR, which is the extended release, it extended the release, as you can see, up to almost 6 hours as 100%. So for us, to have a releasing extent of 6 hours or more would be really a good target in order at least to simulate what is present in the market. However, we want to compete with what is present in the case of the bioavailability and the absorption. So the journey starts. First thing, as we said, the first preparation was the metformin with Juliucer 5013, which is the hydrophobic polymer only. And then we started the variation by changing the carbomer. And as you can see, F2, F3, F4, which stands for formulation 2, 3, and 4, we've kept Juliucer there, but we've now wanted to see what is the addition of the gastroretentive or gastroadhesive is going to really do in the in vitro release of the drug. So in this case, we changed the ratios. As you can see, F4 has the highest carbomer compared to F2. So when we conducted the United States Pharmacopoeia dissolution, as you can see, F1, as we've expected, this is up to 3 or 4 hours, and everything was really released. That's because the nature of the triglycerides of the Juliucer will not really withstand the aqueous environment and will break down. So in this case, it will release the content faster. However, what I can summarize, and for the sake of time, you can see that F2, F3, F4 all has extended the release of metformin to up to 6 or 7 hours. So we know that carbomer alone even has really extended the release before even we have any experimental design changes with regard to the other ingredients. And we continued with the investigations with regard to the variability in those. So let's here look at, for example, how much do I have? 2 minutes? Okay. So I'll have to jump quickly. So this is showing the PEG 35,000 alone without the other content. And you can see here that PEG as a hydrophilic have released the drug much faster compared to the one that has PEG with the carbomer. Again, we've looked at the PEG 35,000 Juliucer, same thing, same trend. And at the end, the PEG 400 as a hydrophilic one. And I want you to pay attention here to those two, which is the F10 which has extended the release to 7 minutes, as well as the F13 here with all those ingredients which really behaved the same. Based on this and based on the XRD, and the analysis and how it really behaved in the in vitro dissolution medium, and as you can see, look, we started with the capsule intact, and you can see this is 1 hour, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And you can see that the components of the tablets has swelled tremendously. So we are as well gaining the size theory, as well as the fact that it was stuck to the wall of the basket during the dissolution. So to sum up this, I have only maybe 60 seconds. XRD, we've characterized all the preparations. We couldn't really get an amorphous components of the image because it's high dose, 500 milligrams. And as a result of that, we managed to reduce the crystallinity. And based on the in vitro dissolution behavior, as well as DSC and the crystallinity results, we found out that F10 as well as F13 were really the best choice to advance for the in vivo study. And as a result of that, now the second step was to demonstrate what we have. Did we really succeed in what we are proposing? So the next step was to do the animal model, and we've used the beagle dog model for oral testing of oral capsules of metformin. And what we have here, 10 seconds and I'll finish. We have the reference, and the reference here is actually the existing XR, or extended release tablets present in the market. Now, all our preparations that we have resulted in actually better bioavailability, with better even T-half and better residence time. Now, the question is, in order for me to claim that it is better than what is existing, I have to take it to the next step. And the next step is actually now to start to trace what is really happening for the tablet inside the stomach. So this is something that is ongoing right now, which is, with the X-ray tracing technology, in order to know how really we achieved this better bioavailability. And I say sneak peek because, again, it's not really part of the presentation because I was talking about in vitro, but I'm here presenting an in vivo result as well. So these are the gangs in the lab, I call them, that I should always, be thankful for their contribution. Mainly, I would like to, other than the funding agents, I would like to thank Dr. Ihab Obasha, who's the Dean of the Veterinary Medicine College in Jordan, University of Science and Technology in Jordan, who really helped us out in getting the Beagle's dog and conducting the in vivo studies in their labs. So, thank you very much. Ready for questions.
Thank you, Dr. Husam, for your presentation. We will open the discussion. Please, 1 or 2 questions for the time.
Yes. Right. True. Well, calling the hydrophobic, you're right. If you look at the Juliusers in general, they are composed of what we call the melting point as well as the HLB value. As a surfactant, it has really, a tendency to form what we call the oil in water. So it will tend to be in favor of the water. However, it's still my description or my characterization of it as hydrophobic because it really made, or it is composed of triglycerides. So these are fatty acids, and their nature is hydrophobic. But you're right with regard to their use. They are used as surfactant. Yes, that's what I'm saying. It is a surfactant, but it's hydrophobic. So this does not negate that. The structure of it is fatty acids, triglycerides, so it is hydrophobic, and it has a high HLB value, which can be really used when you are using it for the emulsion, for example, preparations. You can use it for the formation of the oil and water. And so that does not really negate me calling them as hydrophobic. Did I answer your question? Yes, if you look at that, that's again 5013. Now it goes to the melting point, and this has something to do with the solubility. So it is still, more hydrophilic towards hydrophilic, formation of surfactant, but still it is really a hydrophobic polymer, and it will behave as a hydrophobic moiety.
Okay, I have a question.
Go ahead.
What is the half-life of this metformin pure? And how much this half-life increase or this AUC increased compared to this immediate release and your sustained release? This is one question. And other questions, normally the diabetic patients are used to take this metformin regularly 3 times or 4 times.
Sure.
Say 1.5 gram to 2 grams. So are you aiming to have this once in a day or twice in a day? This type of approach you are taking?
Excellent question. You see, I said it's a sneak peek. I didn't want to share the numbers for the bioavailability study because we did not publish it yet, but I'll share it with you. The regular half-life of the regular metformin is around 8 to 9 hours or 10 hours. We were able to extend it to 14. The AUC was much higher in around 36%. I hope I'm quoting the numbers right. And what you mentioned about the targeting of the dosing, you're totally correct. So if instead of giving 750, that's the medium dose that all the diabetic patients are taking 2 or 3 times a day, if we match the the dose of the 500 to be similar to 1,000 or to 750, then I would really negate maybe sometimes that I would fill it as 750 and give it as once daily rather than really twice or thrice daily. So again, this is going to be with the multiple dosing bioavailability study, and the answer to this would come later, inshallah.
I think this design will take a lot of time and a lot of work is required.
That's why we do research.
Thank you.
Thank you very much. Thank you.